Just a quick overview for Dr. Shea. He'll be leading the webinar today along with Dr. Ray joining us. He'll explain that in a moment. You see a little bit about his work here after getting his bachelor's in science from Duke, in engineering, and he went his master's at Dartmouth and on then to Rutgers for medical residency and so on. You'll see there. Quite pleased to have Dr. Shea today as he's well versed in this whole world of COVID, and not only that, but he believes that Jesus Christ is who he claimed to be, and so as a believer, we share that in common as well, and many people who are joining us from around the country who have found out or heard about this webinar through the organization that's helping host this, which is InterVarsity Christian Fellowship. So John, I think that's enough right now. Unless there's any questions to clarify starting, I'll stop sharing the screen here, and you can, you're up to the plate, John, so to speak.
Great. Well, thank you, Howard, and welcome everybody. I'm very pleased to share this information with you. Hopefully you should now see my my screen. Can you confirm that, please?
Yes.
Great, excellent. Let me just do a couple of things on my end here to facilitate my presentation. So this will be a tag team. Dr. Ray will be presenting after me. My hope is that my presentation will only be approximately 15 minutes. Dr. Ray was initially planned to be here with us live, but there was a death in the family and she had to attend the funeral. And she was kind to actually record her presentation. And so I will be shifting gears after my presentation and putting her YouTube video on. And my hope is that will actually work. So the title of my presentation is COVID-19 Vaccines, FDA Process, Clinical Trial Results, and Real World Surveillance. Here are my disclosures. I have no relevant financial conflicts of interest. I'm employed by the Metro Health System in Cleveland, Ohio. I serve as the Vice President for the health system for research and sponsored programs. I also serve as Chair of the Department of Physical Medicine and Rehabilitation. My academic appointments are at Case Western Reserve University. I am Professor and Chair of PM&R and Professor of Biomedical Engineering. So here's my first slide. This goes through the process of vaccine development under normal circumstances. And so the initial phase is almost purely science, understanding mechanisms, understanding disease process. And the mechanism component of vaccine development almost always happen at a laboratory, typically at a university, and it can take many years, could be decades even at times. Once the mechanisms have been fully elucidated, then an IND or investigational new drug application is submitted to the FDA. And once approved, then a company, a commercial entity, will then carry out clinical trials. And these trials are typically divided into 3 phases. Phase 1 is safety, phase 2 is effectiveness, and phase 3 is both safety and effectiveness. And typically these are done in sequence. Once the clinical trial is complete and the results are submitted to the FDA, called the BLA, or the Biologics License Application. And this is where the approval and authorization process is conducted. Once it's fully approved, then there is what we call real world surveillance, because sometimes what we see in clinical trials isn't always what actually happens in the real world. And you obviously want to see congruency between those two. So some review of FDA terminology. Most of us are aware of something called FDA approval or clearance. And FDA approval and clearance is based on substantial, quite significant evidence. The drug is effective for its intended use, and the benefits of the drugs outweigh the risk when used according to products approved labeling. Investigational treatments are those treatments where scientists are actually still conducting clinical trials to determine if the drug can safely and effectively prevent or treat a specific disease or condition. And of course, these are phase 1, 2, and 3 trials. Now, expanded access is something also called compassionate use. This is a potential pathway for a patient with a serious or immediate life-threatening disease or condition to gain access to an investigational medical product or treatment outside of the clinical trials when there is no comparable or satisfactory alternative therapy. So again, expanded access is for medications that have not been approved yet. It is still under investigation, and therefore it is still experimental. But under this particular pathway, a particular patient may gain access to a particular medication outside the clinical trials environment. Emergency use authorization is the pathway that was taken for our COVID-19 vaccines. And I want to spend a little bit of time sharing with you what this is. So in certain emergencies, the FDA can issue an EUA to provide access to medical products that may potentially be used when there is no adequate, approved, and available option. This is different from FDA approval or clearance in the following way. The FDA makes a product available to the public based on best available evidence as versus substantial evidence, without waiting for all the evidence that will be needed for FDA approval or clearance. The FDA carefully balances the potential risks and benefits of the product based on the data currently available. It is effective only during an emergency declaration, and therefore when the declaration ends, it is no longer effective. But EUAs can be reviewed or revoked at any time during the public health emergency as the FDA continues to evaluate the available data and patient needs. There are some COVID-19 specific EUA requirements. They still need to do a rigorous clinical trial, with very high internal validity design. They must demonstrate safety and effectiveness data. They must show at least 2 months of follow-up after clinical trials completion. The typical approval requires 6 months or longer. And there must be a plan for following up on the vaccine safety, which involves investigating any reports of deaths, hospitalizations, and other serious or clinically significant adverse events. The manufacturer, however, still ultimately must apply for FDA approval once the emergency is over. So let me take you through this schematically. So again, I'm going to just focus on the 2 pathways. One is the emergency use authorization, and then FDA approval. But the FDA approval, let me go through first, okay? In FDA approval, just as a capsule, testing happens first, followed by approval, then production, and then distribution. So again, testing, as you see here, phase 1, 2, and 3, each phase being larger, and again, in sequence. Once the testing is done, then the company would seek actual FDA approval or clearance. Once approval is secured, production of the vaccine starts. And once production is done, obviously distribution. This is a normal pathway. Emergency use pathway is a bit different. This is where testing and production can actually happen at the same time. So you probably heard while the clinical trials were going, companies were actually making the vaccine. Now, this particular diagram suggests that phase 1, 2, and 3 are all sequential, but it doesn't have to be. There can be actual overlaps. Once the study is done, an EUA is secured. And as soon as the EUA is secured, distribution happens right there. And so obviously this pathway is much, much faster. This is another schematic of how things usually happen. So in the normal circumstances, again, you have the Okay, can you hear me now? Okay, so let me start this again. Typically, there's development, IND submission, clinical trials, approval FDA, and then you have production distribution. This takes 15 years or longer, typically. In fact, one of my innovations is it's actually a medical device. I actually started in 1995, and it didn't get approved until 2016, so 21 years. So under normal circumstances, these things take that long. But here is the EUA. The reason why this was so fast is because most of this early phase was already done. Why? Because we already knew about SARS-CoV-1. All the technology for example, the messenger RNA, as well as the synthetic DNA were already done. Only thing missing was a sequence. Sequence was provided, the vaccine was produced, and then clinical trials started. Now, clinical trials usually take a very long time. The rate limiting step in clinical trials is enrollment. But when you're in a pandemic, not a problem. There are plenty of people getting infected. And so as a result, it only took 10 months to 1.5 years total, okay? And so there was no shortcut in this process. We had, again, all these things were already done. Enrollment was very robust. And so these clinical trials were able to be implemented very, very quickly. So that is the process at the FDA and what the difference is between FDA, normal FDA clearance process and the EUA. What I'd like to do now is to share with you the specific data from the 3 clinical trials. The first to be approved was the Pfizer vaccine. This study enrolled 37,000 individuals, 18,000 in each group. And individuals who were vaccinated, only 8 of those actually developed moderate COVID-19 disease, compared to 162 in the placebo group. As a result, this translates into 95% efficacy. Now, these are the adverse reactions, which I will not go over because Dr. Ray will do that. But what does efficacy mean? Let me tell you what it does not mean. A 95% efficacy of the Pfizer vaccine does not mean a vaccinated individual still has a 5% risk of contracting COVID-19. This efficacy statement is a statement of relative risk. And that is defined as 1 minus relative risk. Well, what is relative risk? Relative risk is a risk of contracting COVID-19 for those who are vaccinated compared to the risk of contracting COVID-19 for those who are not vaccinated, those who get the placebo. So those individuals who have been vaccinated, the risk of contracting COVID-19 was 0.000439. Risk of contracting COVID-19 for placebo, 0.00884038. So when you divide those two, that's where you come up with 0.05. So efficacy is 1 minus relative risk, 1 minus 0.05 is 95%. Okay, that's what efficacy means. But let me elaborate further. This means the risk of a vaccinated person contracting COVID-19 is only 5% of the risk of an unvaccinated person contracting COVID-19. Another way of saying that, the vaccine reduces the risk of contracting COVID-19 by 95%. Remember, 5% is 1 over 20. Therefore, we can say this another way. A vaccinated person is 20 times less likely to contract COVID-19 than an unvaccinated person. Or an unvaccinated person is 20 times more likely to contract COVID-19 than a vaccinated person. So I hope saying these things multiple ways gives you a better sense of what efficacy means. Let's go to the Moderna COVID-19 vaccine. They enrolled 28,000 individuals, roughly 14,000 in each group. Only 5 individuals in the vaccine group developed COVID-19, but 90 in the placebo group. This translates to 94.5% efficacy, very, very similar to the Pfizer vaccine. This is a Johnson & Johnson COVID-19 vaccine. They enrolled 39,000, roughly. This is a adenovirus vector, whereas Pfizer and Moderna use lipid nanoparticles. So this concludes the clinical trials results. Let me go quickly to the real world. There are 3 publications of interest, all from the US, that I'd like to share with you. This the first one comes from UCSD and UCLA. On December 2nd, UCSD implemented a low threshold for PCR testing of symptomatic persons, and they mandated that all asymptomatic healthcare workers also undergo weekly PCR testing. On December 16th, both UCSD and UCLA started vaccinating healthcare workers with the mRNA vaccine. On December 26th, UCLA instituted optional PCR testing for asymptomatic healthcare workers. During the period December 16th to the end of the study and February 9th, almost 37,000 individuals had received the first dose, and of these, 28,000 had already received the second dose. And these are the results of this study. Of those individuals, there were 379 individuals who actually developed positive PCR COVID disease. The absolute risk is 1.03, which is significantly higher than the clinical trial. But note that the great majority of these individuals developed their disease within 2 weeks after the first dose. See here we go, where the effectiveness of the vaccine was rather low. After second dose, only 37 developed. So again, if you want to compare it to apples, the clinical trials looked at effectiveness after the second dose. They didn't look at here. Like I said before, the clinical trials was also very different. And why is that? Clearly, these individuals were not fully vaccinated. The moderate to severe disease in clinical trials versus symptomatic and asymptomatic. In other words, in this particular study, they looked at both symptomatic and asymptomatic disease, whereas clinical trials only looked at symptomatic. During that time period, there was a surge in Southern California, and these individuals are all healthcare providers who are younger and increased risk of exposure. But fundamentally, what you should get out of this study is that after the second dose, very few individuals develop COVID-19. It's very consistent with the clinical trials. This is a second study out of UT Southwestern, 23,000 employees. On December 15th, they started vaccinating with mRNA vaccine of all their frontline workers. During the initial 31 days, 14,265 were vaccinated and 8,969 were not vaccinated. Either they never got to it or they chose not to. Of these, you can see the distribution of having one dose versus both doses. It did not indicate how the testing was done. In other words, I don't know whether these are symptomatic or asymptomatic people who developed the disease. But whatever that was, approximately 350 or exactly 350 individuals developed the disease between these two time periods of December 15th and January 28th. Now, this study also coincided with a rapidly escalating number of new infections in North Texas. So these are the results. This is the risk of developing COVID-19. So if you were not vaccinated, we divide 234 cases by these employees. That's how you get 2.61. Partially vaccinated goes down to 1.82, but when you're fully vaccinated, it goes all the way down to 0.05. So again, a very strong result from a fully vaccinated group of people. What's important here is that once everybody was vaccinated, there was a 90% decrease in the number of employees who were either in isolation or quarantine. So again, real life practical results. So again, these results are consistent with the clinical trials. So very effective. The last real world study was published in late March. Almost 4,000 frontline workers in 8 US locations without prior disease. Time period December 14, 2020 to March 13, 2021. 2,956 of these individuals were vaccinated and 994 were not. What's really important in this study is that all nearly 4,000 were tested weekly, regardless of symptoms. And if they did develop symptoms between testing, they got additional testing. So of these 3,950, 205 COVID positive cases were identified, or 5.2%. Again, with respect to collections, 58% were based on weekly, 42% on associated illness. Let's not worry about that, but here are the results real quickly. On the unvaccinated, there were 161 infections. Fully immunized, there were only 3. And this translates to an effectiveness with with respect to partially immunized, they had 8. So what's really interesting about the study was the effectiveness of partially immunized was 80%, is after 1 dose. But again, 14 days after the first dose. This is a pretty impressive effectiveness after only a single dose. After the both doses, goes up to 90%, which is very consistent with the clinical trials. Let me see here. So, conclusions. Clinical trials results, COVID-19 vaccines are highly effective in preventing moderate COVID-19 disease. COVID-19 vaccines are also 100% effective in preventing severe disease and deaths. And the real world results corroborate clinical trials results. So I'm gonna stop here. I'm gonna stop sharing this screen. And I know we're tempted to ask questions at this point, but I think the more important presentation is the next one. So let me go to that real quickly if you don't mind, okay?
Hi, my name is Amy Ray, and I'm
Can you hear her?
Yes.
All right, we're going then.
Physician here at Metro Health in the greater Cleveland area. I am very pleased to be able to present COVID-19 vaccine safety review data for your group, and I do wish to thank Dr. John Shea for the very kind invitation to do so. I'd also like to offer my apologies for not being able to present tomorrow in real time at your session, but I am recording on April the 15th at 11:30 a.m. because of the evolving dynamic nature of the information emerging around the Johnson & Johnson vaccine. I really wanted your group to have the most up-to-date information that I have. As mentioned in my bio, I am the medical director of infection prevention here at Metro Health, and my interest in hospital epidemiology actually even predated my infectious diseases fellowship. I'm doubly board certified in internal medicine and infectious diseases, but my clinical practice is restricted to infectious diseases. I have no financial conflicts to disclose to you. I would like to acknowledge the importance of this Bible verse. Apostle Paul in his letter to Timothy has been advice that I've attempted to adhere as carefully and closely as possible during my scientific studies, and certainly during the course of this pandemic, which is to study to show ourselves approved unto God. And if I were to alter this or addend this in any way, approved and prepared. So I have lived through the Ebola viral disease scare in Northeast Ohio, which was of no clinical consequence, but really honed some preparedness skill sets that coupled with the H1N1 smaller scale pandemic, which occurred with influenza A. So I have been able to rely somewhat on muscle memory in preparedness as we attempt to ascertain as much about this virus, its transmission, its treatment, its prevention, as is possible. And to that end, I will say that I have attended all of the external data review presentations to the FDA for all 3 vaccine products here in the United States. And I've attended every advisory committee on immunization practices discussion as well, so that I can be as prepared as possible and up to date. So in terms of my objectives, I will very briefly review the safety process in vaccine development from a 30,000 foot view, if you will. But the most of my talk will be spent on the 3 vaccine products approved under emergency use authorization and their performance as both in the clinical trials and the real world environment regarding reactogenicity, which is inclusive of local reactions as well as systemic reactions in response to vaccination. And then importantly, I will discuss serious adverse events, both in the clinical trials and the real world environment. The caveat that I would just like to put forth, the way that I've organized this talk is to discuss each product under each of those topics. And by no means do I think that it is scientifically rigorous to compare them head to head. So there have been no trials of head-to-head comparisons. And so that there are differences among the patient population, the time frame of the studies, the locations geographically, and even the methodology with collecting safety data. So I wanted you to be aware of that as I move through these topics. Safety is a very important part of vaccine development along every stage of the vaccine life cycle. And as you can see on the left, there are multiple regulatory agencies and advisory groups, as well as standardized processes in place to ensure very high safety standards here in the United States. And although these steps along the life cycle appear quite linear or sequential, it's important to also note that phase 3 clinical trials, yes, recruitment has been complete, but safety data is ongoing, the collection thereof. And in addition, this slide does not show the emergency use authorization process, which has been sought and successfully by the 3 vaccine manufacturers of COVID-19 under discussion today. In order for full FDA approval, 6 months of safety data is necessary. So I would like to take a moment to compare traditional vaccine development to that of SARS-CoV-2 vaccine development, because undoubtedly you will have questions regarding the rapidity of COVID-19 vaccine development and concerns raised around that enhanced timeline. So there are 3 points I would like for you to be aware of. The first is, because of pre-existing experience with the first 2 members of the beta coronavirus family, SARS-CoV-1 in 2003 causing the SARS epidemic, and the MERS virus, which still circulates at low levels in the Middle East, the immunogen was already identified in this family of RNA viruses. So it was known, even before SARS-CoV-2 was known, for beta coronaviruses, an antibody against the spike protein, which allows the viral particle to adhere to a human cell, was indeed neutralizing. So as soon as the sequence of the spike protein was released on January 7th of 2020, the race was on. So a lot of what was already known about beta coronavirus and beta coronaviral vaccine products was already in place. The second thing that I don't think is acknowledged as much, which I think is probably one of the most important pieces to consider, is the frequency and severity of the outcome. So of SARS-CoV-2 infection being COVID-19 along its clinical spectrum of mild to life-threatening infection. And so when in traditional development, you're looking at prevention of sporadic infectious diseases such as invasive pneumococcal disease, those infections happen very infrequently, but by contrast, if your outcome is prevention of severe SARS-CoV-2, in the United States, along the course of the pandemic, we've had several thousand deaths per day and tens of thousands of infections per day. So the frequency of the outcome measure is an important piece to acknowledge. And then finally, the fact that our government, among other governments, paid for production of vaccine prior to any approval was a financial gamble, but it was not a safety gamble. So that I think there are, and I'm very pleased that this did in fact pay off. So traditionally, this takes the regulatory approval prior to an upscaling of production. And quickly, this is the Pfizer safety monitoring aid for the phase 3 clinical trial. And I just wanted to share active surveillance by the pharmaceutical company, meaning touch base with the patient at predetermined intervals to assess reactogenicity in the early phase and serious adverse events, which typically happen in the early phase. But that data collection for Pfizer for active surveillance continues for 2 years post dose 2. And in addition to active surveillance by the pharmaceutical company, there are passive, well-established surveillance mechanisms in place in the post-marketing surveillance that are ongoing. And for the first time with COVID-19 vaccines, the CDC and FDA have sought an active surveillance methodology in the form of vSafe, which is a smartphone-based app, which is an opt-in for vaccine recipients to report at predetermined frequency reactogenicity and possible serious adverse events. And just as a reminder, an adverse event is any event not present before exposure to the study vaccination, or an event that is present but has worsened in intensity or frequency after exposure. So essentially, surveillance after vaccination is never ending in a passive sense. Reactogenicity is characterized as local reactions, so actually at the site of a vaccine administration. And those symptoms could include pain, swelling, redness, and they can be graded from mild, moderate to severe. The most severe being the grade 4, which would be requiring attendant medical care and potentially even necrosis or exfoliative dermatitis. And the second type of reactogenicity is systemic events. So really the flu-like symptoms that you may be aware of, including fever, headache, body aches, myalgias, et cetera. And those also can be graded, and the definitions are listed for you here on this slide. Now, the measures used in the COVID vaccine trials, looking at reactogenicity, you'll see that all 3 companies used redness, swelling, pain at the injection site. Moderna added as evidence of local reactogenicity, axillary swelling or tenderness. And versus systemic, which you'll see that all manufacturers included the most common systemic side effects, including fever, fatigue, headache. Moderna and Pfizer looked also at reports of chills, nausea. Pfizer specifically looked at diarrhea, and all looked at myalgias. The mRNA product lines looked at the presence of arthralgias as well. In the Moderna phase 3 clinical trial, local reactions were investigated in all participants, including those who received vaccine and those who received placebo. And as you can see from the data below, pain was by far the most common local reaction, and mostly the reaction was graded as mild. You'll note across the spectrum of the clinical symptoms that there were no grade 4 reactions. And what is not being shown to you is the evidence for the local reactions being more common in younger persons as compared to an older age cohort. Now, Moderna did report delayed injection site reactions that occurred day 8 or later after receipt of immunization, which implies a T-cell mediated delayed hypersensitivity. They reported this in 244 patients after dose 1 and 68 after dose 2, but did not further qualify or characterize those reactions. Now, the post-marketing experience with Moderna vaccine has been elucidated a bit further by a group of authors published in the New England Journal in February of 2021, looking at delayed hypersensitivity reactions in 12 patients. And they reported that the initial local reactions and systemic reactogenicity had resolved in these patients, and subsequently the patients then developed these skin findings. So of the 12 patients, 6 of the 12 had no recurrence with the second dose. 3 of the remaining 6 had a similar appearance of a delayed hypersensitivity reaction, and 3 of the remaining 6 had a lower grade reaction with the second dose. So Pfizer chose to investigate local reactogenicity in a slightly different methodology. They used a subset of their vaccine trial participants of more than 8,100 patients. And you'll see from this data on the right-hand side of the screen that pain is at the injection site is again the most common, is common. More than 70, 80% of patients will have pain. But no, there are again no grade 4 reactions. Younger people were more likely than older folks to have pain at the site of injection. And in the Pfizer trial, there was no report of delayed hypersensitivity events. So this is data from Johnson & Johnson as presented to the ACIP committee of CDC. As of today, the phase 3 clinical trial of the Johnson & Johnson product, that data has not been published in a peer-reviewed journal. So in terms of local adverse events or local reactogenicity with the Johnson & Johnson product, the findings did include a common theme in this discussion, which is pain at the injection site. But again, no higher grade reaction. So having shared with you the clinical trial experience with local reactogenicity in the real world as the vaccine products rolled out, as you recall, we began vaccination in mid-December of 2020. This is a publication from Morbidity and Mortality Weekly looking at vaccine safety monitoring in the first month using the vSafe, which is the smartphone-based app. And the findings here are reassuring in that because of the time frame, they had data in a significant cohort of patients who had received both doses of Pfizer vaccine, but only one dose of Moderna, because you'll remember that the EUA of Moderna was achieved after the Pfizer EUA was achieved. So I highlighted a couple of things, which the percentage of patients developing local reactions in the real world is very similar to the clinical trial experience, and the same being true of injection site swelling. Okay. So in summary of local reactions, pain is very common. So you should tell your patients that it's more likely than not that they'll have some pain at the site of injection. Personally, I felt that the pain was slightly worse than my typical reaction to influenza vaccine, but certainly not the tetanus arm. And then there are no, for your knowledge, no grade 4 reactions, and local reactions are not a contraindication to course completion. And the local reactions can be managed simply by reassurance and symptom management. Moving on now to systemic reactions. In the Moderna phase 3 clinical trial, the data is presented here for you. And as a reminder, systemic reactions include the symptomatology of fever, headaches, really flu-like illness with fatigue, myalgias, arthralgias. And so the areas of interest are with what frequency do these reactions occur and how long do they last and what do we tell patients to help them manage. So as you can see, with looking at any systemic reaction, it's again very common to have systemic reactogenicity after receipt of Moderna product. And the most common complaints are headache, particularly more so after the first dose with the second dose, as is true of fatigue and myalgia. The median duration is about 2.9 to 3.1 days. And again, reactogenicity in general is more common in younger patients than older patients due to the robust nature of their immune system. The Pfizer data looking at systemic events, again, as a reminder, they're looking at a subset of their trial participants. And this is, I apologize for the size of this slide. The first half of the top half is patients aged 16 to 55, and you'll see that again, fatigue and headache are the most common and slightly more common with the second dose. And they occurred more frequently in younger patients than older patients and were very short-lived. And so I received the Pfizer vaccine, had no systemic reactogenicity after dose 1, had very short local, as I mentioned, pain after dose 1. But after dose 2, 14 hours later, I had a very flu-like illness, and I actually did have fever, headache, chills, muscle pain, joint pain. So I sort of had the full spectrum, but as soon as it came on, it was gone within about 12 hours. So that's the human immune response to a possible threat. And then it became quite clear to my immune system that no, the presence of mRNA generating a spike protein or portion of spike protein is not a true threat. It's an opportunity to prime and boost my immune response. So again, this is the Johnson & Johnson data as presented to CDC, and we await peer-reviewed publication. But you'll see that, again, systemic events are mild and quite common, and this mirrors their reports of local reactogenicity. So this is the same data that I showed you previously, real-world data incorporating 1.6 million enrollees in vSafe, which is the active surveillance smartphone-generated app, looking at systemic side effects and local side effects. And this time we'll turn our attention to headache and fatigue, myalgia. As you can see, that these were common. Lesser common were nausea, as we saw in the clinical trials. So the spectrum is similar, and the dose response is also similar in that the reports of fatigue, headache, myalgias, chills, fever increased with the second dose. And again, as a reminder, at this point in time, the data was available only for Moderna vaccine on dose 1, but this is what we would expect from a systemic reactogenicity perspective, and there will be further data obviously forthcoming. So to summarize, systemic reactions, these are common, they're mostly mild, and they're typically characterized by fatigue, headache, and myalgias. You'll note that in the clinical trials that fatigue, headache, and myalgias were not uncommon in the unvaccinated population. The systemic reactions are self-limited of short duration, and a systemic reaction is not a contraindication to an additional dose. So typically all that is necessary is management of symptoms should symptoms develop and reassurance of your patient. And again, encourage course completion to achieve the greatest effectiveness, efficacy in the population. Serious adverse events as defined by the Food and Drug Administration include any untoward medical occurrence that results in death, is life-threatening, and you can see the other categories here. And it's important to note that adverse events, the presence thereof does not imply causation. There may be correlation, but, and this point will become important as we discuss the recent turn of events with the Johnson & Johnson vaccine post-marketing experience. So for the sake of time, I will quickly talk through the serious adverse events. This is the Moderna data, and essentially to summarize at a very high level, there were no significant differences between placebo group and the mRNA vaccine formulations. The same is true with Pfizer. This is again Moderna, placebo group deaths 7, mRNA deaths 6, and including a suicide, a head injury. So there was no safety signal in the Moderna data around serious adverse events. And similarly is the case with Pfizer, and I just summarized it here. But the overall rate of adverse events was low, 0.6 to 0.5, which was commensurate with in keeping with the Moderna findings as well. So what about vaccine safety data of serious adverse events for the mRNA products after the EUA approval? And this is showing data from CDC through February 13th, comparing the vaccinated recipients to those unvaccinated with the expected events in that risk interval. And simply stated, for mRNA analysis in this very early phase of the rollout, no statistically significant increased risks were detected for any pre-specified outcome. So briefly, you'll note that you'll deduce that there were no anaphylaxis reports in the Pfizer data, in the clinical trial environment, nor the Moderna. I believe Moderna found one case as they presented, just the day they presented their information to an external review committee of the FDA. Nonetheless, there is a JAMA paper highlighting the rate of anaphylaxis in cases per million doses administered. And just for time's sake and simplicity, these rates are very commensurate and in line with other rates of anaphylaxis after vaccine products. For your convenience, I've included a couple of slides at the end of my talk in order for you to have a reference on how to manage patients with allergies to injection products or vaccine products, and how to think about them and monitor them after immunization. So the topic of the hour is the Johnson & Johnson phase pause of the US rollout. So this is showing you phase 3 clinical trial data, again, that has not been released under peer review in the United States. That's available and publicly available on the CDC website. But essentially within their clinical trial, they had no increased rate of serious adverse events. They did have 5 deaths due to COVID in their placebo group, but 0 deaths in their vaccine recipient group. They did report hypersensitivity, and they reported venous thrombotic events, which I will show to you in more detail in the next slide. But essentially, most patients who had venous thrombotic embolic events had risk factors such as birth control or smoking, prior clot, known hypercoagulable state. There were 14 thromboembolic events in the vaccine group versus the placebo group, which did not raise a significant safety signal in the clinical trial. So when you look at the breakdown of thromboembolic and thromboembolic events in the Johnson & Johnson clinical trial environment, there was a single case of cerebral transverse sinus thrombosis. And yesterday at the ACIP meeting, the Johnson & Johnson reported out on this case. He was a 25-year-old male who developed a CVST 8 days after a dose of the Johnson & Johnson vaccine. He was known to have, or found rather to have, anti-PF4, which is an antibody that has been associated with these findings that I will further describe to you in the next slide in the post-marketing surveillance data. So the hot news off the press is that the Johnson & Johnson product rollout has been paused in the United States as of April the 13th. So as a reminder, this was approved under EUA on February 27th. And as of yesterday morning, about 7.2 million doses had been administered in the United States. Now, many institutions have not had robust supply, either due to constrained production or contaminated production. And in our institution, our Johnson & Johnson has only accounted for less than 1.5% of our total doses administered. But of the 7.2 million, about 1.5 million have been administered to women ages 18 to 50. So the CDC reported yesterday at our ACIP meeting, the first cases of cerebral venous sinus thrombosis detected post-marketing in the United States. And the safety signal, the first case was detected, I believe, March the 19th. To date, there have been 6 cases, all in women ages 18 to 48, actually. They were diagnosed day 6 to day 13 post-vaccination. All 6 had associated thrombocytopenia and all had anti-PF4 antibodies. So while clotting, such as deep venous thrombosis, is not, is fairly common, it is very unusual to have cerebral venous sinus thromboses with thrombocytopenia. So the incidence rate of CVST, for simplicity, is not incredibly well known, but estimated to be about 2 per 15 cases per million of the population. The caveat and the thing that's quite concerning to myself and many others is that this is a specific kind of CVST. So it's not a common phenomenon, and it can be quite severe. In fact, one of the 6 cases was fatal. So I agree with the FDA decision to pause, and I then subsequently agree with the decision by the Advisory Committee on Immunization Practices to pause as the data set is more complete. And what I mean by that is the first half of the 7.2 million doses, enough time has gone by to hopefully detect all the cases in the first batch or first half of the administered doses. More time is needed to make sure that we have a full understanding of the implications and the case count in the United States population. But this incidence rate is concerning, and the severity is concerning, and it is prudent that this decision was made. And again, it's not a cancellation of the J&J product. There are many good things about the Johnson & Johnson product. The efficacy against COVID-19, the 1 dose as opposed to a 2-dose series, and the stability of the product have been very important to reach some underserved areas, including homeless populations, homebound populations. So I think that while I agree with the pause, there will be detriment to the rollout, but in the United States, we have other products, Pfizer and Moderna, that are doing the, are more commonly administered. So I think that we'll have soon more data in the next week to 10 days to further define the use of Johnson & Johnson in the United States. So in summary, the vaccine safety process in the United States is robust, as evidenced by this experience with the Johnson & Johnson. This is exactly the way the system is supposed to work. The VAERS system and some of the other systems are long-standing and tried and true, and the addition of some active surveillance is important in my view. You should know that reactogenicity, both local and systemic, is common, but usually mild and short-lived. And overall, the safety product of the vaccine, safety profile of the COVID-19 vaccine products in the United States is strong. And I'm excited for Dr. Che to share with you the excellent efficacy in preventing severe COVID and COVID-19 deaths. With regard to the Johnson & Johnson formulation, I anticipate that we may see something of which has occurred, some version of which has occurred in the European Union, meaning the AstraZeneca vaccine, which is also a non-replicating adenoviral vector vaccine, has similar findings of CVST with thrombocytopenia and anti-PF4 antibody. And because of the patient profile in which those events occurred, in Europe, the AstraZeneca vaccine is being used in an older population who have not been demonstrated to be at risk. That remains to be seen here in the United States, so stay tuned. I anticipate data on an hourly, daily, if not hourly basis. Thank you very much for your kind attention, and I would be happy to come back and take some questions and answers anytime. Thank you. Be well.
Okay, so thank you so much for joining us, and I already thanked Amy for going the extra effort to record this. And so it is 4:58, so I regret there isn't a lot of time remaining officially, but I am willing to stay on to try to answer any questions regarding the efficacy as well as adverse events. If I can't answer the question about the adverse events, I'm happy to deliver that to Dr. Ray and get her response.
John, we had an early question. I asked people to post from Rosalind. It asks, what are the risks for women who want to become pregnant in the near future? and followed up by another question. Go ahead.
So that particular question was raised by two individuals, and both of them are actually in Europe right now. The reason why that question became more prominent is because one of the individuals was vice president of Pfizer. This is something that's been in the internet a lot. It's been promulgated by those who have strong anti-vaccine feelings. There is a particular protein in the system that if it's impaired can indeed impede pregnancy. However, the connection between the vaccine is actually non-mechanistic. There's no reason to believe that the vaccine itself will have any impact on this process. In fact, there have been plenty of pregnancies actually during the clinical trial. And so this has been fact-checked multiple times, and there is no scientific basis for this information that's been promulgated.
All right, good. The follow-up, and this is, I don't know if you can take this or not, regarding, I just finished my second shingles shot. I'll get the COVID-19 shot on April 21st. Should I use the opposite arm?
I don't think it really matters.
All right. Next question is posted here, and then we'll get to others who would like to ask. You can ask it, just unmute yourself after this one. Our friend of ours in her 50s had a mild paralysis reaction on one side of her body after receiving the first Pfizer shot. It lasted on a short while, gone in a day. Naturally, there's cause for some concern for her second shot. Any thoughts?
Yeah, so any of these, technically, these will be called anecdotal events. This particular event may have been associated with the vaccine itself. What I would do at this point, so here's a theoretical connection. So because of this concern about thromboembolism or thrombotic event, it is very possible that the vaccine could create a situation where a thrombotic event would happen where a cerebral artery, not the venous system, could have been affected. If that's the case, this may be a TIA, transient ischemic attack. Now, it's possible it's from the vaccine. It's also possible that this is something that this particular person should check out regardless of the vaccine. Because if this is a TIA, even independent of the vaccine, this individual may be at risk for a full-blown stroke, even independent of the vaccine. So my recommendation is to actually follow up with a primary care physician and would need additional evaluation, including carotid ultrasounds, echocardiograms, as well as possibly a CT scan.
Okay. A couple other questions that are posted, John. I don't know if you can see them. Do you know the efficacy and safety of Sputnik 5 vaccine used in Europe and some other countries? Yeah,
there's a lot of discussion about that, but I haven't studied that enough to be responsibly answer that question.
All right. And prior to that, if a 60, 65-year-old healthy and active adult contracts this virus, what's the percentage rate of survival? I've heard about 90 to 95.
So the overall survival is quite good, actually. And so that's probably, if you contract it, that's probably accurate. I say your survival might be even higher.
Okay. Can you tell us about the anti-PF4 antibodies?
So that only thing I know about that antibody is that, and Dr. Ray mentioned, this particular CVST is not your run-of-the-mill CVST. In other words, CVST happens. And by the way, the incidence rate of CVST could be as high as 15 per million, which is much higher than the incidence that we see in this clinical trial, which is 1 per million. But because associated with this particular antibody, and there's also, she mentioned thrombocytopenia, which is a decrease in platelets, that this may be a very specific event related to an immune response from this vaccine. That's probably the best I can say. It's probably the best indicator of specificity of the effect and not a spontaneous event.
All right, I'm going to open the floor here. If you want to unmute yourself and ask a question, go right ahead.
There was a question that I see here with Brazilian and South African variants. Will there be a need for a new vaccine later on? So far, the South African variant is pretty resistant. And so all the vaccine makers are actually working on adjustment of their vaccines to address this. So that is correct. So right now, the UK variant appears to be pretty well covered by Pfizer and Moderna. I don't know the information with respect to J&J.
Marilyn has her hand up. Go ahead, Marilyn. Hey, unmute yourself.
Okay, I got it. Hi. I'm 84 years old and I had bad symptoms, flu-like symptoms after my first Pfizer shot. And my doctor said maybe I already had antibodies. So I wonder if I have to have the second shot.
That's a great question. Here's some data, information. Older individuals tend to have less of a reaction, in part because their immune system is not as active or not as robust. We do know that if you actually had COVID, then your first dose is in all likelihood will give you a very, very strong reaction as if it was your second dose. So that's the basis for your primary care doctor saying that. But unfortunately, we don't know for you specifically if that is the case, because there are plenty of people who have very severe reactions after the first dose without having had COVID. And so I don't know what to tell you. I guess what's good is that according to the most recent data, the efficacy after the first dose is still pretty good at 80%. And
good.
I'm not going to give you any medical advice over Zoom.
I know, but I just wondered, is there a way to have an antibody test before I have the next shot or not?
I don't believe that is readily available for the public.
Okay. That was my question, and thank you so much.
You're welcome.
A couple other questions there. Do you see them, John?
Yeah, let me go down.
From Matt?
If a person has already had COVID, do they need to get vaccine? At this point, the recommendation is yes.
Okay.
Just be aware that you might get a strong reaction. Let's see. How long do you think we will be monitoring COVID and wearing masks? Well, I think it depends on how well the public behaves. Once we get the herd immunity, where 80% plus are immune, then I think we're going to be in good shape. Now, I think at this point, I agree with Anthony Fauci that by the end of summer, we might be in fairly good shape. Now, remember, there is a recommendation from the CDC. If you are fully immunized, you could do exactly what you want to do. You don't have to wear a mask when you're interacting with other people who are immunized. You can hug them. You can do all that stuff. Now, the recommendation is not to, but when you're outside in public, though, that you would still wear a mask. And so, as soon as we as a nation become, reach herd immunity, that's where we're going to be. My hope is in the fall that I'll be able to go to medical conferences again, without having to even wear a mask and so on. So the future is very difficult to predict. It's a hazardous business. In the Old Testament, they had a habit of killing those individuals who got it wrong.
Yes, indeed. John, Ida would like to ask a question.
Hello, Dr. Shea. This is Ida Halasqua from West Virginia University. Thank you so much. This was very, very nice that you summarized that for us. I have a question about some molecular mechanisms or action, like when we get the vaccine, in the arm. Both, if you could elaborate a little bit both on the mRNA and the adenovirus synthetic DNA vector.
Yeah.
When
So
when it gets to like cells, like whether the muscle cells are the ones or just the antigen presenting cells that do the work of producing the spike protein. And I'm very curious, and I don't know why this question keeps popping in my mind, that whether the cells that are producing and spitting out the spike protein to which the immune system reacts, how long they stay in our body, and if those cells divide, and if they carry this mechanism with them producing.
Okay, so I think the best thing for me to do is refer you to our prior webinar, where we actually went through the mechanism of mRNA as well as DNA vaccines and how they work, and it also addresses the question of does this thing replicate? And these viruses are non-replicating viruses, and so you do not have this virus going on and on and on. And so if you want to refer, in fact, Howard, if you can just share the prior link, there's I think a fairly good comprehensive presentation of the physiology of how these vaccines work. Would that be okay?
Okay, thank you.
You're welcome. There's a question, how is herd immunity determined? It's based on, so if you have COVID, then you're considered part of that, you're contributing to herd immunity. If you are vaccinated as well. And so when you count, basically you're counting everybody who's been immunized as well as those who have been vaccinated. And then you divide that by the entire population, and that's the percent that is fully immunized. The real question is that for COVID-19, what is the appropriate herd immunity percent? The reality is nobody knows. The guesstimation right now is if 75 to 85% of the US population is immunized or had COVID, most specialists opine that that will be sufficient for us to go back to quote normal unquote. There's another question, would not children need to be immunized to create the herd immunity numbers needed? The answer is yes. And that's why all the manufacturers are testing the vaccine with younger ages. Okay, I think I gotten everything on the chat box.
All right.
Okay, well, Howard.
Well, so in summary here at the end, just to repeat, we have the recording we've done for today. It'll be available on a link that we'll have in a couple of days for you. If you would like the copy, just send me an email request, and Bach has put that up for everybody. My email, if you scroll back up into the chat box, you'll see my email address, or you can also send this request to Bach as well, bach.chu@InterVarsity. He's much more efficient than I am, and I would highly recommend that. So, well, that takes us to the end, and thank you, Dr. Che, and thank Dr. Ray for us as well when you chat with her next. And I'm going to pray as we close. Lord Jesus, thank you for this time together, for the opportunity to know something and to find out something and to do science, to research something, all of which you've given to us, and we don't take it for granted. Thank you for our minds as well as our hearts. And take these things now, Jesus, and cause them to multiply in us the knowledge that of you and about you to others. In Jesus' name, amen.